Hello, my name is Gyula Cégény. I'm a research fellow at Department of Plant Biology, University of Pécs, and I'm reporting now on behalf of our research group. Within the frames of EPPN 2020, we applied for a transnational access project to be implemented at the sunscreen facility of Helmholtz Centrum Munich. In this study, we investigated whether solar UV radiation can support different bell pepper cultivars' acclimation to disadvantageous cold temperatures. As an important agricultural plant, capsicum varieties are grown all over the world, in warmer countries as well as colder areas. In Europe, the sowing usually happens in early spring, then it takes at least 10 weeks until harvesting. Due to the possible unfavorable weather conditions across Europe, the first two steps usually take place in greenhouses, plant nurseries, or plastic foil tents, which conditions are mainly UV excluding. Then the farmers plant their seedlings outdoors in mid-spring, when the unfavorable conditions are still can be sources of problems. To prove that, it is well enough to check the temperature recorded last 2 Aprils in Pécs, Hungary. Even in the middle of the southwest region of Hungary, springtime temperatures can be really cold. So, in collaboration with Dr. Jana Barbara Winkler, we designed a transnational access experiment to simulate this environmental challenge of capsicum seedlings under isolated circumstances. We tested the cold tolerance of the following 3 capsicum cultivars in Helmholtz Centrum Munich. The first one is called Szegedi, and it is one of the most popular cultivars in Hungary. It has strong green leaves, but its development is a bit slower than the rest of the seedlings. The second cultivar is called Amy, also a Hungarian one. It is developing faster than the other two, but its leaves are pale green, therefore it might be sensitive to various stress factors. The third one is called Black Olive, and the seeds were kind gifts of Vertilum Company. Black olive name is coming from the dark purple phenotype caused by the accumulation of anthocyanins. The involvement of that cultivar in the study was happened because it may has altered responses to UV at low temperatures. Prior to the experiment, Dr. Winkler and her group were pre-cultivated the seedlings in a sunscreen chamber under the climatic conditions of the control treatment. On the day I arrived, 5-week-old seedlings were organized into 2 groups. While seedlings in the first group were kept under control conditions, the other group was exposed to simulated sunlight for 5 days. Then the seedlings were further organized into 4 groups at that point of experiment. Therefore, we've had UV pretreated and non-pretreated seedlings in both control and cold chamber. The cold exposure of the plants was 5 days long as well. During the experiment, we characterized the dynamics of plant growing by 2 CCD cameras. Seedlings were put on a rotating platform, and the 2 cameras have recorded 9 images of each plant. The kinetics of pigment accumulation in different leaves has been also monitored. We did that by daily recordings of the pigment contents with the Dual-X instrument, which is able to estimate the chlorophyll, flavonoid, and anthocyanin indexes based on the leaves' optical properties. Beside the pigment analysis, light acclimated photochemical yield parameter of particular leaves was also investigated by using a Waltz Minipem instrument. The phenolic and carotenoid pattern of the leaves are also going to be analyzed by analytical methods. At the end of the report, I'd like to thank for the EPPN for conducting this study, which would not have been possible without the support and the transnational access. In vivo recordings were done by Jana Barbara Winkler and Petra Seibel from Helmholtz Centrum Munich, Eva Hideg and Gyula Cégény from University of Pécs, and the HPLC analysis will be done by Kristóf Csepregi, also from University of Pécs.